Hello, biology students. Mr. Wagner here, taking you through our DNA Model Activity Lab. We're going to be using DNA Model Activity A today. DNA Model Activity A should be available for you in your folders from your teachers. And it's very important that we're going to put this together with some pieces of things. So the kids that are in class, they're actually going to be doing this with model kits. You unfortunately at home do not have model kits available. So what we're going to do is we're going to make do with the next best thing. I've got here some pieces of licorice. I've got some gumdrops of different colors. And I'm going to use this as my model kit. We want you, even though you can't be in school, to do something actually with your hands, something manipulative, in which you're putting together a model of DNA. You don't have to use licorice and gumdrops. You could be using something like Legos. You could be using something like pieces of pasta or black olives, anything like that you might find around the refrigerator. Maybe you're using crackers. Maybe you've got a couple different kinds of crackers or whatever it is. If nothing else, you're just going to take blank pieces of paper and you can cut up stuff and color and write on the blank pieces of paper and lay it out together on the table. The plan is that you're going to take a video of what we're doing where it says show your teacher. We're going to show the video of you, and you're going to answer the questions that are on these sheets here for this DNA Model Activity Lab. We're going to talk about what DNA is, how it's made, its structure, how it replicates, and then also finally how it makes an amino acid. So tell you what, let's get started. Let's read what it says for DNA Model Activity A. You're going to put your name at the top, the number of the kit, that's for the kids that are in class. You will not be in class. But it says this project requires you to use pre-made modeling pieces, or in this case at home, your own modeling pieces, and this paper to complete the activity and receive a grade. Do not lose any of them. If pieces are lost, you'll be deducted points from your lab score. You don't have to worry about that. You can use whatever the pieces are you put together on your own. It says, first of all, take 8 white tubes and connect them with 9 black 3-prong pieces that you see below. All right, so we're not able to do that because we don't have the pieces, but here's what Mr. Wagner's going to do. I'm going to take a toothpick, and I've got some pieces of licorice that I cut in half. I'm going to use these for the black pieces. In fact, I'm going to line them up right here on my sheet. These are my black pieces right here. And for the white pieces, again, color is really not that important, I'm using parts of a Swedish fish. So I took a little Swedish fish and I cut them up into pieces so I'd have enough. And I'm going to use the Swedish fish, SW, for the pieces right here. And I'm simply going to connect them with a toothpick. Going to show you exactly what that looks like. So let's start here. Let's take this one, put a toothpick through here, and push it down if I have to. Tap it down a little bit. There we go. Go all the way to the end. Whoopsie, came right out again. There we go. And let's take a Swedish fish. We're just going to poke it through here. There we go. Careful not to poke my fingers. And then another piece of licorice. Poke it down if I have to with the table. There we go. And another Swedish fish. All right, it looks like it says I need a total of 8 of the Swedish fish and 9 pieces of licorice. So tell you what, I've already got some already together just to save a little bit of time. Here it is right here. Let's see if we can poke them together. And let's see what we got. Let's make sure we got it correct. I got licorice, Swedish fish, licorice, and it says I should have 9 of the black pieces. Black ones, remember, are my licorice. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. I need one more. I thought Mr. Wagner miscounted. So let's go one more for 9. And let's put this one here. There it goes. And a little piece of a Swedish fish. Put that right through there. Best I'm able. And stick that right in there also. Okay, it doesn't have to be neat. Doesn't have to be fancy or anything. But it says here that the white tube, that would be the Swedish fishes, they represent the phosphates, and the black one, in my case the licorice, represents the deoxyribose sugar. So I got deoxyribose sugar, and they're connected by these phosphates. 1, 2, 3, 4, 5, 6, 7, 8, 9 of those. And it looks like I need 8 of the Swedish fish. 8 Swedish fish, again, would be 8 phosphates. 1, 2, 3, 4, 5, 6, 7, 8. Okay, I am all set with this piece. Now, going left to right, add the following nitrogen bases. This is my ACGT. A and T go together. Let's make a little line so we know that they'll go together. So we got A and T go together. And then the C and G go together. C and G. And the way I remember that is a capital A is straight lines, capital T is straight lines, C is curvy lines, G is curvy lines. The straight line ones go together, the curvy line ones go together. Okay, and Mr. Wagner's going to use gumdrops for these. And I had to have a change in color since I didn't make black gumdrops. I went to yellow. It's okay. So come up with some pattern on your own. It's really important that you write down whatever this sheet is where your different color codes are. So I have adenine is going to be green, cytosine is yellow, guanine is red, thymine is blue. And then for the uracils, I'm using white, just like they have. So I only had to change one. Okay, so here's what it says. I've got this one here, and I want to follow this code right here. This code right here. It says starting left to right, I want an A. So I got to find an A. It looks like my A is green. So I'm going to take one of these that's green, and I'm going to poke it on just like this. Let's see. So here it is. I'm going to put this one right in here. That hooks right onto this one. There it is. I think I'll use a half one next time. In fact, you know what? I think I'm going to break this one in half. Make it a little bit smaller. That way I have less toothpicks to worry about. So let's break this one in half. There we go. All right, that'll be a little easier now to work with. So I got this one right here. There we go. That's my first one. My A was my first one. My A was green. There it is. Next one, it looks like it says a C. C is going to be yellow. Let's put that one in here. C is going to be yellow. There we go. And the next one then is a G, which is red. Let's use a red one. Break a toothpick here. Use a red one. And you're welcome to stop the video as we go. In fact, I'm going to stop and work ahead a little bit so you can catch up. So I want you to see what we're doing. A, C, G. A is green. That's my first one. C is yellow. That's my next one. G is red. That's my next one. I'm going to pause the video and finish up. You go ahead and finish up as well, and we'll check back in here in one moment. All right, Mr. Wagner is back. I've just put a few more in here. Let's see what we got. I've got my last one here. My last letter is a G. I looked down on my chart. G is red. So I'm going to use my little red gumdrop, put that through here, and connect it to the last one. Remember, we're going from left to right, and let's double check our letters. It says A is green. C is yellow. G was red. It looks like A is going to be green again. There it is. Yep. T is going to be blue. I'm using purple instead of blue, but it's close enough. G again is going to be red. The next one is a C, which is a yellow, a T, which is a blue or purple, and finally the G, which is red. So I've got all of them all set just like this. Notice how it looks down on my sheet? Colored tubes are there. I got the colors all sticking out. Okay, so that is great. If you're in class, we're having our teacher check it at this point. But since we're not in class, we're not going to do that. We're flipping over to our next page, and it says now what? Now, twist the twist tie prepared around the upper. Okay, we don't have that twist tie prepared, but we're going to do this. It says, this which marks the section of the DNA transcribed by the RNA in part 2. Okay, so I'm just going to leave it like this. So we'll talk about that in part 2. It says now use 8 more tubes and 9 more 3-prong to create the other side of the DNA molecule. The other side of the DNA molecule, just like this. And what I want to do is I want to line them up so that they match with the ones that they match with. What do we mean by that? Well, Mr. Wagner already made one to save a little bit of time. Let's see if we got this right here. Let's see if we got this right. Here's my other piece that I made. So it looks like this one. And notice again that I've got my 1, 2, 3, 4, 5, 6, 7, 8, 9 deoxyribose. And I got my 8 Swedish fish in between them. There it is. And I've already put these on that match up with the one that it matches up with. What does that mean, matches up with the one that matches up with? Let's take a look. Remember what we said when we match. We always match up. A matches up with T. That's my green and my blue always match up together. So guess what? Here's the first strand I made. It's got green. So I've got to match up one which is blue. Blue's got to match up with that. So I made another piece and I put on here now the blue that matches. It looks like my yellow is my cytosine. And it looks like the cytosine matches up with the red, which is guanine. So look at that. Everywhere I got a green, it matches with a blue or a purple. Red and yellow and red. Red and yellow, green and blue. Blue and green, red and yellow, yellow and red. Blue and green, red and yellow. So they always match up, every single one of these. Excuse me. Everyone matches up with the correct colors. They match up correctly. Green and blue go together. Green and blue. And yellow and red go together. Now let's see what it says in the next step. It says now that I've made those that match up, just like on here, I'm going to connect them together. So let's go ahead and connect them together just like this. This one goes right in here. Might be a little bit awkward here if they don't fit exactly, but that's okay. It's okay to make mistakes in science. If you're in my class, you know that. It's okay to make mistakes. That's how we learn things. Scientists make mistakes all the time. Let's see if we can put this one in here. See if this one fits in here. Notice it broke. That's okay if it breaks. Not the end of the world. We just want to make sure that you know exactly what we're doing when we match these things up, that they go the right way. We're not grading you really on neatness or whatever. We just want to make sure that it works. And yeah, it's a little crooked. Notice it's crooked. It doesn't really fit exactly perfectly right. The point is that I know that these two go together, these two go together. A and T go together, C and G go together. That's the most important aspect. It says now, connect the two halves of the DNA using it. So I'm connecting them just like this. So this is actually what a DNA molecule looks like. Just like this. And if we twist it, I don't know if it can handle the twist, but that's what it would look like with the shape. Okay, twisting that ladder like that shape. Let's answer some of the questions here. You can go ahead and answer these questions. Twist the shape of the ladder. What do the white pieces represent? What do the black three-prong pieces represent? And in yours, you're going to put down whatever you're using. For me, I'm going to put down what does my licorice represent? What does the Swedish goldfish represent? And then write that in there. What do the uncolored plastic pieces represent? Put that in there also. Answer that question as well. And then you're going to send us a video of what this looks like. Twist it a little bit. If it breaks, don't worry. Put it back together. But that's your first part, making a DNA molecule. Part 2 is going to be transcription. Transcription. Let's see what part 2 looks like. This is now where, see, we've got this one. Yep, got that one. Yep. Part 2 is transcription. All right. Part 2 says this. Now that you know what DNA looks like, let's see how it sends information to other parts of the cell. Create a single strand of mRNA. Remember, mRNA is only single-stranded. So I'm going to pause the video, and we're going to make exactly the same thing we did. 8 white tube pieces and 9 of them. So another one of these whole sections. I'm going to pause the video and make that while you make it. Okay, Mr. Wagner, back again. Look what we made. We made another one of this mRNA strands. Notice the difference is it's only one-stranded, not two-stranded like this. So here's what I got. Looks like I got my licorice again and my Swedish fish or my phosphate. Remember, the licorice represents, of course, the deoxyribose, and then the phosphate is the red one, and it matches up just like that. So I got to have a total of 9 of these licorices. 1, 2, 3, 4, 5, 6, 7, 8, 9. It's sticking out at the end. That's okay. It's not the end of the world. We'll make it look nice. All right. Now it says, using the proper mRNA transcription technique, add the appropriate colored tubing to the mRNA molecule. Okay, let's see how we do that. Well, what happens is, in transcription, this molecule actually, the DNA pulls apart a little bit. And when it pulls apart, like this, let's see if I can get this together. It separates or pulls apart. So it gets unzipped. Unzipped, just like this. There we go. So it unzips. Zip, just like a zipper. You see how a zipper kind of looks? So that's why it kind of works just like that, like a zipper. And what we're going to do now is we're going to copy this strand from mRNA. Let's say that this section is a section that talks about a certain kind of protein. This is going to be a protein. So what we're going to do is we're going to take it and unzip it, and mRNA is going to come in and copy this protein down. Let's see if there was a one that I had to check as far as which one it's going to be. So it looks like we're using the ACG. Okay, so yep, I had to make sure which one I'm using. I'm going to use the one for the copying, which is the AGC. That's this one at top. That's this one right here, because the A was green, The C is the yellow. So I'm going to use the top one. Notice I didn't move them. I'm going to temporarily take this one out because it unzips. So here's the section of DNA that just unzipped, and then I want to copy. So now mRNA is going to jump in here and start copying down exactly what this code is. Notice that when it copies, it's going to use uracil, those are the white ones, instead of T. So instead of the blue ones, wherever I'd match up a blue one, like here and here, instead now I'm going to match up a white one. I'll do the first one and show you exactly what it looks like. Get a couple little toothpicks here. And let's see what this one's going to be then. So the first one looks like it is a G, because G is green in my sheet right here. G is green. Give you a little bit more sheet so you can see it there. So normally, green would match up with blue, but remember I don't have blue. I don't have thymine in RNA. So instead, let's do a different color. So instead, sorry, I got a camera on a pivot here. There we go. So instead, this one matches up now with C. This is RNA. RNA, it matches up with U. A matches up with U in RNA. And this one, of course, is for the DNA. DNA. And this one also over here is my DNA. There we go. Okay. So let's see what we got. We got to match up a white now to this green. So let's take one of these, poke it through here, and it's going to match up right here as a white one. That's my white one that goes with my green. Next one is a C. C was my yellow. Yellow can still match up with guanine. We didn't change anything for that one, so it's still going to be red. So let's make this one red. Match up here. All right, so you see how we're doing it now, right? The only difference is I'm using the white uracils instead of the blue or purple T's. All right, so I'm going to finish this and come back in the video in 1 second. I'll let you work on it as well. You're welcome to pause the video anytime you want. Okay, Mr. Wagner's back again. Again, this is my RNA, my RNA. This is the DNA that was unzipped, and I'm copying it down. The only difference is everywhere I have a thymine, which was my purple gumdrops, I'm going to use a white one, which is uracil. So those match up, green and white, yellow and red, red and yellow, green and not blue, not purple, but white. Here I've got a purple, but that's okay because that's in the DNA. That goes in DNA. This is my RNA. It just doesn't go in RNA, that's all. So that one still matches up with the green. So that would be my thymine still matching up with A, which is adenine. Red and yellow, yellow and red, purple and green, and yellow, red and yellow again. They match up just right. This is my RNA code. So what happens is RNA comes in here, the messenger RNA, copies down this code, and then leaves. And then the DNA molecule zips back together, and it goes back to the way it should be. And now I have over here my mRNA, which was copied from the DNA that zips back together. Now I've got the code, like breaking into the Hot Pocket layer and stealing the recipe for the Hot Pockets so that I can make some amino acids. Now with this recipe, I can build my amino acids. All right, let's take a look at what it says here. So have we got our sheet here? It looks like we've done that already. We've copied this was our DNA. This is the mRNA code. All right, let's go on to the next part. Transfer RNA. Brings it to the ribosome in order to create the polypeptide. That's a fancy word for the protein chain. So now it says attack a black amino acid to the blue tRNA. Do this 3 times for all 3 amino acids and RNA. Okay, let's see what we got. We're going to put this together here. So it's going to look like this. I got one of these, and I got my piece. So I've got this one. Excuse me. Got one of these, and then another one of these. And then one of these again. I don't know if I'm going to have enough. In fact, hold on a minute. Mr. Wagner's going to try something different. Okay, Mr. Wagner's back. I had to use a different piece of licorice. We're going to use this one. That's okay. It does not have those in between, but that's all right. Not the end of the world. And what we're going to do is, this is my messenger RNA. This is translation now. We're going to put this into, this is the code. Now we're going to put the code together with the pieces that belong to it. So if this is the white one is uracil, remember. Let's get the other sheet up here. That way we can just double check. The white was the uracil, and that matches up with A. So on this translation piece, I'm going to have an A, which is green. So let's do that. Let's put this one here. Here's my green. And that matches up with that one. Green and white match up. The next one's going to be a yellow. Yellow, of course, is going to be my cytosine. And cytosine goes with the red, which is guanine. So I'm looking at my code. It's very important I have my code. That goes together. And here's a yellow, which is a cytosine. Over here, then, is going to be a guanine. Over here is a guanine. Let's put this together. There it is. And this one goes here. Okay, they always copy in triplets. Always copy in triplets. So those go together. My uracil and my adenine, my white and green. My red guanine and my yellow cytosine, and my yellow cytosine and my red guanine. So this is, it gets fed through. This literally gets fed through the ribosome like this, kind of like a dollar into an ATM. And then what happens is it comes up, copies this one, got it. The next one comes along and copies the next set of 3. I've already done that in advance. Here it is. Again, notice how they match up exactly right. That's my next set of 3. And then finally my last set of 3 match up. Again, red and yellow, guanine and cytosine, and my adenine and my uracil, and then, of course, my cytosine and my guanine again. Check. There it is. So this was the code. I copied the code, and I made my translation RNA. Each one of these is going to represent, as a triplet, they're going to come together, and they're going to make an amino acid. Each one of these is a base. Let's go on to our next part then, part 4. It says use the codon table. Let's move this forward a little bit here. So I'm going to get rid of my mRNA. I've got my 3 amino acids. Here they are. That's the ones that I just made. So amino acid 1, 2, and 3. There it is. And it says use the codon table on the next page. That's this one. And using this codon table, we're going to find out which these are. So follow along with me. You've got the table there. Let's take a look and we'll figure out how to read this table and write down what the amino acids are going to be. So here's my first amino acid. I kept them all in nice, neat order. So let's see. This first one looks like a green. Green is an A. So I'm going to write it down here. I've got A. Yep. After A, I've got a yellow one. My yellow on my chart, of course, is my C. And then my red on my chart is my G. In fact, I'm going to copy all these down while I got it right here. My green again. Green is going to be my A. That's going to be my uracil. And finally, my red is going to be my G. And let's copy down this last amino acid. The yellow, of course, is my cytosine. The white is my uracil. And the red, of course, is going to be my guanine. So there's my 3. That's my first one, second one, third one. There it is. Now let's find out what the name of this amino acid is. So here's how I read this chart. Okay, I start with my first base. It says first base over here, and I'm going to pick one of the boxes. So it looks like my first base is an A. First base is A. Makes a little bit of room, sorry. Get a little bit messy here, not enough room. First one is an A. So I go here, there's my A is. There it is. First base is A. Next one is a C. Here's my second base. So let's take this whole row over to C, and I'm in this whole area right here. This is my A, this whole box. This is my C, this whole box. So I kind of got this whole box area. This is A and C, all of these. Now I go to my third one, which is G. I come over here to my third base, and I find out where G is. There it is. And I follow it across, and then I circle this one. This is threonine. T-H-R, threonine. Threonine. There it is. See how I found that one? Threonine. I went this one first, the whole box, this one, covered all those boxes, and find out what the third one is. Let's do another one. We'll do them all. So let's find out what this next one is. The next one is going to be a green. The green is going to be, of course, an A. So here's my first box, A. I'm going to use a different color for this one. I'll use blue. And now my next box is a U. There's my U. So I'm in this box right here. I'm in A. This is my A. And I'm in U. U, U. I'm in this blue box. Now my third one is a G. I come over to G. Here it is, G. And it looks like I have this one right here. There it is. I'm going to copy it down. Methionine. 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 Yep, there it is. And you know what? We got another worksheet where we're going to be doing a whole bunch of this. So I'll do the last one. Let's do it together as well. You stuck with me this far? We'll go ahead and help you with the last one. We'll do a black this time. C. Going to come up here to C. There's my C. Going to go over to the U. There's my U. It's going to be in this box right here. See that box? There's my C box and my U box. So I'm in this box right here for C and U. And then my last one, I come over here. I find where the G is. There's my G right there. Come across. Looks like it's going to be this one. Leucine. 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 All right. So that now is my 3 different amino acids that are come together to make a protein. So that's how mRNA copies from dRNA and makes this protein. The DNA strand had the ingredients for how to make it. mRNA had to copy it. There was my copying piece. And then it had to translate it to these 3 little sections. Each one of these is amino acids. They come in triplets. That's why there's 3, 3, 3. And then there's a name for my special amino acid. Right there. Let's see what else I got. Do I have one more sheet on the back? Something to do on the back? Let's see. Nope, that's the other page, part 2. There was part 3 and part 4. There's my chart. Here is my chart right here that I just used. And now it says part 5. All the parts of the DNA RNA project must be disassembled. So if you're in class, you have to disassemble those. Otherwise, you can just take these apart and eat them. Please, if you do eat them, make sure you don't choke on any of the toothpicks. Very, very important. Take the toothpicks all the way out, if that's what you're using. Again, maybe you're using Legos, maybe you're using pieces of paper with color, whatever it might be, but make sure the toothpicks are out so you do not kill yourself. All right, great job. Make sure you send the video of you putting all this together. You can watch the video again if you need to put all this stuff together to submit your lab to your teacher. Mr. Wagner's here. Have a really great day, everybody. Take care, and God bless.